This is a little lecture on section 13.2. Has to do with the structure of DNA, and basically getting into the structure of DNA. There is a lot of history that goes along with that, and I am going to send a little YouTube video to go along with tomorrow's instructions. It'll kind of go through a little bit of that history. I think the history of this is actually pretty neat. It's pretty informative. So overall, what is the structure of DNA molecule? Well, up until the time we talked about Avery and all of his associates that worked on the identification of what the molecule was that was actually causing inheritance. Hershey and Chase experiment with bacteriophages added to that. So basically where we're at right now in the 1950s is where we know what's causing heredity. We just don't know how it's doing that, how it's passing traits on from one generation to the next. And that's kind of where we pick up in section 2 here. DNA is going to be made up of individual units called nucleotides. Now, if you remember back on chapter 2, we talked about organic molecules and how they were polymers, and how each of the polymers were made up of monomers, which were individual units put together like Legos to make one big structure. And that's how DNA is. DNA is one of the 4 major organic molecules that we studied in chapter 2. DNA is made up of monomers called nucleotides. Okay, so hopefully that makes sense to you. Carbohydrates are made up of monomers called simple sugars. Proteins are made up of monomers called amino acids, and so on. So the polymer DNA is made up of individual structures called nucleotides. And each one of those nucleotides is going to be made up of 3 other individual molecules. And you need to know that. Each nucleotide is made up of a 5-carbon sugar, and that's called deoxyribose. We're going to see another type of nucleic acid that we'll talk about later on. It's called RNA. And the only difference between deoxyribose and ribose is that deoxyribose has 1 less oxygen atom, which is why they call it deoxyribose. So it contains a 5-carbon sugar, which explains the little pentagon shape in the nucleotide, the gray nucleotide shape, or sugar shape, a phosphate group, which is indicated by a small circle and a P on the diagram, and a nitrogen base. Nitrogen bases are going to be adenine, guanine, cytosine, and thymine in DNA. There are 4 kinds of DNA nucleotides, okay, and that's going to be based on the kind of nitrogen bases that they have. You can see that each one of these nucleotides is going to have a sugar and a phosphate, sugar, phosphate, but each one of them will have a different nitrogen base. Some of the nitrogen bases are going to be large, 2 of them, adenine and guanine, and they'll be called purines. 2 of the 4 different kind of nucleotides with different nitrogen bases are going to be smaller, and they're going to be called pyrimidines. That's the cytosine and the thymine. As we'll see later, a small one will always bond with a large one, and adenine will always bond with thymine, and cytosine will always bond with guanine. So we'll always end up with the same width in the middle of the DNA ladder. So we have adenine, guanine, cytosine, and thymine. So each nucleotide is going to be determined or named by its nitrogen base because the other 2 components, the sugar and the phosphate, are the same. So the backbone, or the sides of the DNA ladder, is going to be formed by the sugar and the phosphate group, and then they'll each attach to the nitrogen base, which will make make each nucleotide. The nucleotides are going to be joined together, and the sequence that's put together could be any sequence. Okay, that's what makes up the message that's on DNA. It's the sequence that we put these nucleotides in the DNA. There was a very famous scientist from California. His name was Erwin Chargaff. And Chargaff, along with a lot of other scientists who were studying nucleic acids, made a very important discovery shortly before Watson and Crick actually put their structure of DNA model together. And that was that the percentages of guanine and cytosine bases are almost equal in any sample of DNA, and conversely, the percentages of adenine and thymine bases are almost equal in any sample of DNA also. So what this gave Watson and Crick is a clue as to how the molecules fit together in the DNA model. They always knew where they found a guanine, they would also find a cytosine, and conversely, where they always found an adenine, they'd find a thymine. Adenine never bonds with anything other than thymine, and guanine never bonds with anything other than cytosine. So that's a very important clue as to how the structure of DNA is going to be put together. There was another big piece of evidence. Now, when they were trying to construct this molecule and see how everything fit together, there were a number of scientists who were coming up with different models, and that in itself is very interesting history. All the different models that had come up and then come and gone because of new evidence that kept coming up with scientists that were studying DNA. One of the key things that happened was a bit of work that was done by someone called Maurice Wilkins and Rosalind Franklin. This in itself is a little bit of an interesting story because Wilkins, who was per se in charge of the study, was working with Rosalind Franklin, and indeed, as it turned out later, Rosalind Franklin was doing most of the work and Wilkins was taking more of the credit. As it turns out, in most biology books now, Rosalind Franklin gets credit for the X-ray diffraction studies that were done on the structure of DNA, and Maurice Wilkins' name has disappeared. So there you go. Sign of the times. We're coming out with information that is kind of interesting. Rosalind Franklin, though, unfortunately, after she did her studies, was not given credit for this because she died before the structure of DNA came out and the news of how useful her studies were to the structure of DNA. Bottom line is Wilkins and Franklin used X-ray diffraction studies to get information about DNA. And basically what they did was they bombarded the molecule with X-rays and recorded the scattering pattern of the X-rays on film. And basically what we're doing here is they bombarded it. It's like bombarding an object with a bunch of BBs that had ink on them. And then as the BBs came off and they scattered, they would leave an ink spot on a piece of paper. And you could kind of get an idea of what the picture was or what that structure looks like by how the BBs diffract and the X-rays diffracted. Another way of looking at this is like looking at a shadow. If you were from Mars and you didn't have any idea what a bicycle looked like, and all you got was the use of a shadow, you put a bicycle up against a wall and you looked at the shadow of the bicycle, it would give you a very good indication of what a bicycle would look like by the way that the light scattered against the wall. And the X-ray diffraction studies basically, and this picture you're seeing here is the famous Photo 51, which gave eventually Watson and Crick a very good idea of what the actual shape of the molecule was. You can see that it crosses and gave them the idea that the molecule that they were trying to study, the shape of the molecule, was probably two strands wound around each other in like a ladder, like a twisted ladder form in a double helix. So using the clues from Franklin's pattern, James Watson and Francis Crick built a model that explained how DNA carried information and could be copied. Watson was a young scientist in his 20s when he started these studies. Francis Crick was a little bit older, had studied mainly physics and a little biology in his time, but was quite a bit older than James Watson. He was an Englishman, and Watson went together with Crick and studied at Cambridge University in England, and they sat and they pondered the structure and spent a lot of time going through the various ideas on what the structure of DNA could be. What they came up with using Franklin's pattern and using Chargaff's rules for pairing, base pairing, Watson and Crick created a model that was what was called a double helix, a twisted ladder, in which two strands were wound around each other. Actually, when we look at this, there are two individual strands, but they're in opposite directions. When we put this model together, we can see that it kind of looks like if you had a rubber ladder and you grab both ends of the rubber ladder and you twisted them in opposite directions. That's exactly what a DNA model would look like, where the sides of the ladder would be alternating units of the sugar and the phosphate, and the rungs or the steps of the ladder would be where the sequence of nitrogen bases bonded together would be. For instance, an adenine and a thymine together here, and a cytosine and guanine together here. Watson and Crick discovered that hydrogen bonds can form only between certain base pairs, adenine and thymine, and guanine and cytosine. Now, if you remember, hydrogen bonds aren't like covalent bonds or like ionic bonds. They're not quite as strong, so they can be easily broken, but they do hold molecules together. Whenever we talk about the nitrogen bases bonding together to make the steps in the ladder, adenine always bonds with thymine and guanine always bonds with cytosine. And we call this pairing of the nitrogen bases base pairing. So we always know that on one side, if there's adenine, the other side will have thymine, and on one side has cytosine, the other side will have guanine. One of the other things that is mentioned in this section is kind of the fact, where did he go from there? First of all, Watson and Crick, although they did put together the structure of the DNA model, they really didn't know how it worked, okay, at the beginning. Watson and Crick, along with Rosalind Franklin and Maurice Wilkins, got the Nobel Peace Prize in 1962 for their study of their structure of the DNA, which pretty much everyone started to agree with at the time. But although their double helix model explained a lot of the aspects of DNA, it had the limitations of not being able to tell them initially how DNA actually carried the hereditary information, and it didn't explain how the cell could use that information to make traits actually come about. So, and that's what we're going to be talking about in future sections, how the DNA model carries a message. We'll do that in the following sections. And that's all for now. Adios, amigos.